All right, so we like to start off with a little bit of data. I'll go a little slowly, I guess, let some stragglers come in. So we like to start off with a little bit of data. So we had some expectations for yesterday, how it would go. And we thought it would go pretty normally, but it turns out it went even better than what we could expect. So I want to thank the participants for great questions, great engagement during the tutorials, and I also want to thank Liberty for a great talk and a great introduction to our workshop. So today we've got two sessions, as we did yesterday. So everyone is here for session 3. We have 3 talks in this session. And then immediately after, not immediately, after a break, we'll have the tutorial session, and you should have received links for that if you're invited to the tutorials. Let us know if you didn't receive a link to the tutorial if you were meant to. And don't forget, one thing I want to mention is tomorrow we have another talk, Giacomo, who is out of our field, but is going to tell us some important lessons that we might benefit from about data sharing and standardization. And we also have a discussion session tomorrow. So if you have any questions that might fit in with the discussion or a Q&A tomorrow, please add them to our Slack channel. We have a channel called Questions for Day 3 in our Slack. I'll put the link into the chat when I'm done. But today we have 3 speakers today: Narayan Sankaran, Michael Thornton, and Joshua Kulasingham. So I will turn it over to Narayan to share his screen and tell you a little bit about him. There we go. So now we have Narayan Sankaran. Narayan is a postdoc fellow in Eddie Cheng's lab. So we are 2 for 2 on speakers from Eddie Cheng's lab. There at UCSF, he uses intracranial methods in neurosurgical patients undergoing treatment for epilepsy. He studies the neural computations and specializations that underlie music perception in higher order regions of the human auditory cortex. Before he went to UCSF, he completed his PhD at the University of Sydney in 2018. He was supervised by Simon Carlisle, and his PhD was entitled The Structure of Cortical Representation of Speech and Music, big music guy. So today he's going to tell us about 2 different projects that he's working on that demonstrates decoding and encoding approaches and how they can help us to understand the neural mechanisms that enable us to perceive and appreciate music. So without any further hiccups, I'll turn it over to Narayan. Thanks, Narayan.
All right, thank you very much. Can everyone see my slides okay?
Yep.
Okay, great. So thanks so much for that introduction, and thanks for the invitation to speak today to all the organizers of CNSB. This is a real honor, and I'm really excited to talk about this work. So over the course of the past day, you've probably heard about a bunch of encoding and decoding approaches, maybe for investigating the neural processing of speech. And what my work is essentially doing is using similar approaches to study the neural processing of music. So I'd like to start by just showing you a simple piece of music. And this is just a short melody that's indicated by the notation at the top here. And just as we would do for speech, this music can be characterized in terms of several important properties or features that may be represented in the brain. So I'm just going to play this melody for you so you can get a better intuition here. Okay. And so, for example, some basic acoustic features we could consider is we could look at the amplitude envelope, which is this solid black line that's tracing the waveform of the sound. And we could also consider the spectrotemporal representation of music, which tells us how power is distributed across frequency bins and across time. But we can also consider some more perceptually relevant and meaningful representations of music. So for example, we could look at the absolute pitch of each note in the melody. And of course, this is perceptually important because it'll determine how high or low a given piece of music sounds. But then beyond just absolute pitch, we can see that between the adjacent notes in a melody, we have these kind of discrete pitch changes. So this is what we refer to as intervals. And we measure these in semitones. And this is an important feature as well, perceptually, because even if we shifted this melody up or down in absolute pitch, as long as we preserve the pattern of intervals, we would have no trouble recognizing the same piece of music. Okay, and so the features I've described so far, we could think of as low level in the sense that they reflect information that's within the immediate or the short-term acoustics of the signal itself. But when we listen to music, we also perceive things like tension and resolution, things which ultimately induce emotions. And so we wish to consider these sort of processes too. And so one important feature related to this higher order experience of music listening is what's called the tonal hierarchy, which basically describes the perceived stability of each pitch in a melody. Another way we could capture this sort of tension and resolution is by modeling the expectation or the surprisal of each note, which I'll talk about later. But for now, the main point about the tonal hierarchy is that it reflects the fact that in music, not all pitches are perceived as equal. So instead, we have this hierarchy of perceived stability. So in this figure on the right here, on the x-axis, we have the 12 distinct pitches that exist in Western music. And this is behavioral data. So listeners were hearing each of these pitch classes, and they were rating the perceived stability of each within a musical context. So let's just focus on the solid line. And what you can see is that there are some pitches that are perceived as highly stable. And so these play important structural roles and occur more frequently in music. And so we give them special names like the tonic and the dominant, while other pitches are relatively unstable. So they would sound like they don't fit within the musical context. And so together, this behavioral profile is what we call the tonal hierarchy. And it's really fundamental in shaping our higher order perception of tension and resolution. Okay, and so it's also a higher level feature in the sense that the information isn't readily available from looking only at the short-term acoustics of the signal. Instead, it requires that listeners have some prior experience or are familiar with the structure of, in this case, Western tonal music. Okay, and so in a nutshell, the overarching objective in studying the cognitive neuroscience of music is to study how an acoustic waveform arrives at the ears and gets progressively transformed in the brain into more and more meaningful and emotion-inducing musical representations. And so as with speech, the way we study this is by looking at either the encoding of these various stimulus properties in the ongoing brain activity, or by doing the inverse and using decoding models which map brain activity onto stimulus features. Okay, and the type of questions that we might be interested in asking can broadly fall under these categories. So during music listening, we can ask what relevant stimulus information does the brain actually represent? We're also interested in knowing the temporal dynamics of processing. So when are these different types of information represented in the brain relative to when they occur in the signal? We're also interested in where in the brain it's processed. For example, is there some interesting spatial organization where different features are represented in different parts of the brain, for example? And then lastly, can we say something about the specific encoding mechanism that represents this information? So how exactly does activity within a given region or a given neural population vary as a function of a feature such as pitch? Okay, and so today I'll talk about two different projects that use decoding and encoding approaches to examine these questions. The first uses MEG data and tries to decode musical information from MEG activity. And because this is non-invasive and it's recorded at the scalp level, we don't have amazing spatial resolution, but we do have good temporal resolution. So we can ask what information is represented, and we can particularly focus on when that information is represented. And then in the second part of the talk, I'll talk about more recent work that involves applying encoding models to intracranial activity. And because this is ECOG data now, we have good spatial and temporal resolution. And so we can really ask all four of these questions. Okay, so for the first project I'll talk about, we were really interested in the tonal hierarchy, because even though it's so fundamental to music perception, we really know relatively little about its neural basis. And so firstly, we simply asked whether the patterns of cortical activity and MEG activity represent the tonal hierarchy. That is, do they represent the perceived stability of pitch? And then if so, we asked when is it represented relative to lower level representations like the absolute pitch of notes. Okay, so we presented MEG participants with natural music. This was a natural music stimulus, and so they heard all 12 of these discrete pitch classes within a musical context, and we had many different observations of each pitch class while we were recording activity. And then this results in a neural data matrix that has dimensions of time by MEG channels by observations. Okay, and one crucial pre-processing step is that we need to perform some sort of dimensionality reduction. So note that our original data consists of 160 MEG channels, and we want a way to reduce this down to something more manageable, both for the computational load and also so that we're not overfitting our decoding models. So luckily for us, many of these channels may just contain noise, or they may be correlated with one another and therefore contain lots of redundancy. And so an effective way of reducing the number of dimensions is using principal components analysis, or PCA. It essentially looks for covariance across the different MEG channels, and it'll linearly weight them to produce these new principal component time series that are statistically independent, so they're completely uncorrelated. And typically, the first few PCs will explain the majority of the variance. So in our case, we were able to reduce the dimensionality of our data set from 160 channels to just 20 PCs while still retaining 99% of the variance. So now, rather than working in the original sensor space, we were classifying or decoding activity in this principal component space. Okay, so now we're ready to apply multivariate pattern analysis, or MVPA, to decode the pitch class that listeners heard from their corresponding MEG activity. So the idea is fairly simple and perhaps familiar to many of you, but let me just illustrate with a simple example. Let's say we want to decode the activity corresponding to two different pitches, pitch A and pitch B. And so what we do is we take their neural activity and we divide the data into training and test sets. And then we'd use the training data to train a linear model. In this case, we used linear discriminant analysis. So conceptually, it's very similar to standard linear regression, except that the LDA finds a linear combination of the input features that best separates out these two classes. So this defines what's called a decision criterion, which is this red dashed line. And in this kind of simple schematic, we're just visualizing this in 2 dimensions here. But remember, we have 20 principal components. So you'll just have to use your imagination to think about this in a 20-dimensional space. And so we take this decision rule that we've learned from training, and we then apply it to the independent set of unlabeled test data, and we see how well it can predict the class of pitch. And so the key conceptual point to remember is that the accuracy with which we can discriminate between the activity of these 2 different pitches provides an estimate of how dissimilar their neural codes are. Okay, and what's more is, because we had good temporal resolution, we were able to train and test a new classifier at each time point. And so this results in a curve of classifier accuracy across time. And looking at different landmarks in this curve, we can track the flow of stimulus-related information in the cortex. Okay, so we applied this entire MVPA pipeline for every pairwise combination of the 12 pitch classes in our stimulus set. So now let's look at some results. So in these decoding plots, time 0 corresponds to the onset of notes, and a chance level of accuracy is 50%, or 0.5. So we see that before the onset of notes, performance was at chance, which is just a nice sanity check. And one simple hypothesis is that when we hear a note, when we're listening to music, our brain simply represents the pitch or the fundamental frequency of that note. And so if this were true, then the ability to decode two different notes should just correspond to the difference in their F0 value. And we can see that during an early period from approximately 100 milliseconds to 250 milliseconds after the onset, that's exactly what we see. So for example, the accuracy for decoding notes separated by large F0 differences, which is this blue curve, is greater than the accuracy when notes were more proximate in their F0 value, which is this red curve. Okay, but then what's really striking is that from about 200 milliseconds onwards, we found that the distinction between notes in the brain really reflect the differences in their perceived stability as detailed by the tonal hierarchy. So here in this panel on the right, the three decoding curves are discriminating between cortical activity of notes with different levels of separation on the hierarchy. So for example, in the green curve, classifiers are discriminating between neural activity of the most stable and the most unstable notes. And we see that performance is relatively high. And in contrast, if we look at that purple curve, this is comparing the unstable notes that are all situated at the bottom of the hierarchy with one another. And we find that their neural activity is the least discriminable. And so these results together suggest that when we listen to music, there's a brief period soon after the onset of notes where cortical activity represents the fundamental frequency, while later activity reflects their perceived stability. And so this is something that we confirmed and characterized in more detail using representational similarity analysis. So if you're interested in this analysis, you can ask me at the end, but in the interest of time, I'll move on for now. So in this first part, we have some answers to what kind of information the brain represents during music listening. For example, it represents the F0 and it represents the perceived stability of different notes. And we've also been able to say something about when these different types of information are represented as we listen. Okay, but of course, there are many unanswered questions. For example, we didn't specify where in the brain this information was being represented. And we weren't able to characterize the specific mapping between the stimulus features and neural activity. And so to get at these questions, we now turn to ECOG. And so these are direct recordings that are taken from the surface of the human temporal lobe in patients who are undergoing clinical monitoring for their seizures. And so each ECOG electrode, there's a schematic of this on the right. Each electrode is able to record the activity of a local neural population. So this is about 50,000 neurons. So we presented 8 different ECOG subjects with about 25 minutes of natural instrumental melodies while recording. And we focused on this region, the superior temporal gyrus, which is a higher order region in auditory cortex, which we already know is a hub for complex sound processing. But beyond the question of whether this region is simply activated when we listen to melody, we still understand very little about the specific information encoded there. And so here we asked whether, neural populations in the STG are tuned to 3 very specific critical features of melody. Okay, so the first 2 features are what I already described. We have the discrete pitch of each note in the melody, and we have the intervals that are formed by these adjacent pitch changes between notes. But for considering the higher level structure now, our analytical framework doesn't require that we have 12 discrete classes of pitch. And so instead of using those behavioral ratings of perceived stability, we can use a more powerful and context-dependent and continuous measure to capture more accurately that pattern of tension and resolution that we experience in music. And so specifically, we can use a pre-trained statistical model of music to measure how unexpected each note in the melody was conditioned on the notes that came before it. So this is something that we call musical expectation. And the key idea is that these musical sequences aren't random, but they instead have some statistics that govern how they're likely to progress. So in this example melody, you can see that the third to last note, for example, is relatively unexpected. And that's because it violates the established tonality and also the melodic pattern that was established previously in the phrase. So we have these three fundamental dimensions of melody: pitch, intervals, and expectations, that each contribute to our perception in different ways. And we basically wanted to find out how local neural populations in the STG encode these three features. And of particular interest, we asked whether each of these different dimensions is encoded by the same neural populations or by spatially separate ones. Okay, and so here we're just looking at the neural activity of a single ECOG electrode. And we've aligned the activity so that time 0 corresponds to the onset of notes. So we identified electrodes such as this one, which were modulated by pitch. In this case, you can see that it's tuned to relatively low values of pitch. But we can also see that its activity remains relatively unchanged across different intervals and levels of expectation. And then in a different set of STG electrodes, such as this one, in the second column, we found tuning to intervals. In this case, it's tuned to descending intervals, but not the other two features. And then finally, really interestingly, we found electrodes that were positively modulated by how unexpected a note was. So the more unexpected it was, the stronger the response, but not by the pitch or the intervals of these notes. And so the response patterns at these three electrodes indicate tuning to a distinct property of melody in each case. And so what this suggests is a spatial code in the STG for representing these different dimensions. And so to formally test this hypothesis, we used temporal receptive field modeling. And I know this has been a focus of a lot of the workshop, so I won't go into too much detail about the methods. But in a nutshell, this is our model formulation. We're saying that we can model the activity at each electrode at a time point t as a linear combination of the different melodic features and at different lags plus a residual error term. And importantly, when we do these models, in addition to the three melodic features of interest, pitch, intervals, and expectation, we also want to statistically control for the variance that's driven by other properties by including many different acoustic predictors in the model. So we trained our turf models using 80% of the data and using ridge regression. And then once we have the turf weights from training, we use it to predict responses to the held out 20%. And when we evaluate these predictions against the actual ECOG response, that gives us a model R² value for each electrode, which is what you see plotted on the brain for one of our participants on the right-hand side here. And in general, you see that these models are pretty good. So the best models are able to explain up to 40% of the variance in the continuous neural activity. So an R² of up to 0.4. But of course, what we're really interested in are these three melodic dimensions of pitch, intervals, and expectation. And we want to use this turf modeling framework to look at the encoding of these dimensions in more detail. And so what we need to do is we need to use a variance partitioning approach. And so specifically, by computing the variance that's uniquely explained by each of these three features, we can quantify the extent to which each electrode encoded each of these features. Okay, and so to do this, we find the unique variance explained by a feature set J. What we do is we have the R² value that corresponds to the full model that contains all the predictors. And we can also find an R² value of a reduced model that omits the feature J but contains all the other predictors, okay? And then the unique variance explained by that feature J is simply the difference in the R² value between the full model and the reduced model. And so we did this for each electrode and for all our participants. And here I'm just showing the results for one participant. So the size of these pie charts that you can see indicate the magnitude of the unique variance, and the color indicates the feature that was encoded. And by doing this, we can observe some pretty interesting things. For example, you can see there's some spatial organization along the SDG. So the most anterior aspect seems to be primarily tuned to intervals, while this more posterior region is more tuned to expectation. And then perhaps more importantly, consistent with those evoked responses that I showed you earlier, we see that for the most part, each electrode's activity is largely explained by one single distinct feature of melody. So this is just for one participant, but when we pool across all our participants, we can see the same trend in this scatter plot. So if, for example, the pink electrodes that are distributed along the x-axis had unique variance explained by intervals, and these are largely non-overlapping with the green electrodes along the y-axis that were tuned to expectation. And we see that the same is true for the other two contrasts. So pitch versus expectation in this middle scatter plot on the right, and pitch versus interval on the bottom scatter plot. So in each case, we see these kind of independent dimensions of encoding. So using this variance partitioning approach, we can make the claim that pitch, intervals, and expectations are encoded within separate populations in the STG. Okay, and then finally, we can also confirm this by looking at the receptive field weights. So I know you've seen these sorts of weights before. And here we're showing just 3 different electrodes that were tuned to each of the 3 features. So for example, the weights in the first column correspond to an electrode that had unique variance explained by pitch. And in line with this, we can see that the weights are primarily concentrated over pitch and not the other 2 features. And in this case, you can see it's kind of monotonically tuned to respond more strongly to lower pitches. And then the same is true for an electrode tuned to intervals in the middle column, where you can see the strong tuning to ascending intervals. And then lastly, the weights for an electrode tuned to expectation in this right-hand column, where you see monotonic tuning, where a more unexpected note drives a stronger response. Okay, so just as you've probably seen for speech, this shows that we can use temporal receptive field modeling, and it can be really useful for answering some of the same questions about music. And the one result I showed you today is that these different perceptually relevant types of melodic information are represented within separate populations in the STG. Okay, and so I want to leave time for questions, and so I'll just thank all of you for listening, and I also want to acknowledge all my collaborators. So for the MEG work, I worked with Bill Thompson at Macquarie University in Sydney, and Tom Carlson, and my supervisor, Simon Carlisle. And then for the ECOG work at UCSF, I worked with my PI, Eddie Chang, also Frederick Dunison, and Matthew Leonard. So thanks a lot.
Nice. What a great talk. Thanks, Narayan. Got quite a bit of time for some questions. We have a couple in the Q&A session. I don't know if you want to take a look at that, or sorry, the Q&A window. Fazla asks, why not linear classifier an LDA? Is it only for low computational complexity or any other reasons? I found non-linear SVM performs better than linear. That's why I'm curious.
Yeah, so we tried using SVMs and we were essentially seeing the same result. What I like about LDA is just the interpretability. You get a really clear set of weights and it's easy to interpret what those weights represent. So yeah, we just stuck with as simple as possible, basically.
Yeah, you can move down if you've got them pulled up. You can just answer them yourself.
Sure. Okay. So Anna is asking, were the ECOG electrodes placed bilaterally? If so, were there any differences in the representation patterns between left and right SCG? Yeah, so we had 8 participants, 5 of them were left hemisphere, 3 of them were right hemisphere. So not a huge amount of data in the right hemisphere, but we basically saw tuning to all 3 features of melody in both hemispheres bilaterally. So no significant hemispheric differences in the data. Which is kind of, I mean, maybe from the literature, we might expect to see more tuning to these features in the right hemisphere, seeing as they're all kind of relating to pitch. But interestingly, that's not what we see. Okay, and then David, are these separate populations representing different temporal scales? So, okay, I think I understand what that question is getting at. So the bulk of the variance in activity that we're explaining are these kind of transient bursts in activity that follow notes in a melody. So there is like phrase level structure and kind of other broader temporal scales to consider when we listen to music. But because of this stimulus set that we used, where we had these brief melodies that were only a few seconds long, we're really interested in the kind of note by note activity. And so the temporal scales are all pretty similar for these features that we're looking at.
Cool, I have a question.
Sure.
Actually, I have a couple if no one else has questions, but so my first question is related to how you decode pitch. I'm curious, what are the features of MEG that you use to decode pitch? Do different channels respond differently to different pitches? So you showed that one example of an electrode tuned to pitch. I was wondering if it was tuned to that specific pitch. It looked like it was around 200 hertz. Or does it respond differently? Or do you need to take advantage of the tonotopy in cortex, whatever is there under the electrodes? So are you able to decode pitch with one electrode, basically?
Yeah, that's right. So maybe in primary regions, there's some evidence that there's kind of periodotopic maps. So there's kind of essentially the equivalent of tonotopy for complex pitch. Yep. But what we see in the STG and in these higher order regions is actually there's more of like a linear response where a given neural population will adjust its firing rate monotonically with pitch. And so what you actually get is a full representation of the complete range of pitch at one electrode. And so an interesting question following from that is, why do we have multiple pitch electrodes throughout the cortex? Why is there so much redundancy? And that's something that I don't know yet.
Seems like a good question to ask.
Yeah. So
there's another question in the Q&A, and it looks like Nate and Joe have their hands up. We'll see if any of the participants. Do you see Ed's question?
Oh yeah, okay. So I see his question. Yeah, so I think basically he's correct in his answers to his own question. So intervals is just the note by note difference. It's just the semitone difference between adjacent notes in a melody. And then expectation is, yeah, we're using a pre-trained statistical model of music. In this case, it's a LSTM, so it's a type of recurrent neural network. It's exactly right, it's the magenta model. So this is trained independently on thousands of Western melodies, and we're predicting how surprising a given note in a melody is, conditioned on both the pitch and the duration of the previous notes that came before it. Another option here would have been to use something like the idiom model of expectation, which is just like an n-gram model of music. The problem is those kinds of models aren't as good at capturing the kind of long distance dependencies that you have in music. And something like an LSTM is better able to do that, which is why we went for this. And I see that Ed Layla also asked, yes, all the music we used was monophonic, so just a single melodic line.
Cool. Michael, do you want to ask a question? You can unmute.
Yeah, that was really cool. I hadn't heard of this note stability idea before. I was wondering, firstly, how is it what criteria are used to define the note stability? So what are people asked in order to come up with that score? And the second question was, is this a thing, if it's known, is this a thing that people kind of acclimatize to or develop, or is it a thing that people are kind of born with? Thanks.
Yeah, those are great questions. So the answer to the first part of the question is, this is kind of a classic result from the early 80s in music cognition, where they basically presented a brief musical context. So it was just like a series of chords that unambiguously established a musical key, and then they would play what's called a probe tone, which is just each of the 12 different pitch classes. And the listeners are asked to rate what they call in the paper the goodness of fit of the pitch with the context. The term that's developed in the literature for that is stability, or tonal stability. And so what you see is that the tonic is the most stable, followed by the dominant, and then the median, and then all the way at the bottom of this hierarchy, you have the kind of non-diatonic tones. So it's kind of the canonical profile of how stable each of these pitches are in a musical context. It comes from these behavioral ratings, but it's also intimately connected to the statistics of natural music. So the most stable tones are also the ones that occur the most frequently. Yeah, and then, sorry, I think I've forgotten the second part of your question. Could you just repeat that?
Yeah, it was basically, is this, I think maybe you started to answer it in your previous answer, but is it related to, is this something we're born with or something we kind of develop?
Right, so I mean, the main piece of evidence that says this is culturally acquired, it's learnt, is that the tonal hierarchy is by no means universal. So different musical systems around the world have a completely different tonal hierarchy. Of course, there are some kind of innate acoustic components that lead people to claim that things like the tonic and the dominant have natural relationship to one another. But for the most part, it's generally accepted that listeners have to implicitly learn the tonal hierarchy through exposure to music. Yeah.
Okay, great. Thanks a lot.
Cool. I want to give Nate the opportunity to ask a question since he's a music dude.
Okay. Hi. Yeah, this was really cool. So in the MEG data, you were talking about how the interval and tonal hierarchy were represented at different time scales in the neural response. And I'm wondering if you saw something like that in the ECOG data, where the expectation responses later than interval responses.
Yeah, so that was definitely something I predicted, that we would see like an early F0 representation in the turf weights and then a later kind of surprisal response in the turf weights. But actually, that's not what we see. So the latency of the encoding in ECOG is all kind of equivalent between the three features. One possibility is that in the MEG data, we're picking up on whole brain activity. And so there are different regions that are representing F0 versus these kind of higher order perceptual aspects of musical pitch. Whereas in ECOG, we're just focusing on the STG. And again, there's this question of why we even have a representation of F0 in the STG, because by that point in the kind of processing hierarchy, it's kind of pretty redundant. I mean, it's represented all the way from the cochlea onwards. So yeah, I'm not entirely sure, to be honest, why we don't see latency differences, but those are a couple of potential reasons. Another thing is we downsample the ECOG data to 100 hertz to make it more manageable computationally. And then we're also extracting the analytic amplitudes. We're doing the Hilbert transform. And so you actually have a slightly acausal signal when you extract the Hilbert. And maybe for that reason, we're kind of smearing the latency differences that we'd expect to see between the encoding of absolute pitch and the encoding of these higher level features. That's just another possibility, but yeah, I'm not sure.
Thanks.
Cool. Joe, did you give up on your question? You want to ask it?
Yeah, I didn't want to stretch the time, but just a quick question on more primary areas. Just to know if your data confirms what I saw in the past, like that there is this transition between hashless gyrus to TTS to then planar temporal, and where this sensitivity to expectation emerges. But for example, in hashless gyrus, I didn't really see expectation there, but just at the anatomical transition there, it was emerging. Did you see something like that as well? Yeah,
I wish we had data from Heschel's. Unfortunately, we don't. But you do see that there are these kind of electrodes that are strongly tuned to expectation, and they are pretty close to where Heschel's would be. But again, these electrodes are not in the sulcus; they're remaining on the lateral surface of the gyrus. So unfortunately, I don't have an answer for you.
Okay, fair enough. Thank you.
Yep.
All right. Well, if there's no more questions, I just want to say thanks, Narayan. Everyone, give him a thanks. Talking silently.
Thank you.
Great. I'll turn it over to Stephanie, who
Yes.
will introduce our tutorial teasers, I guess we could call them. I just want to emphasize that even though these are going to be a little bit of context for the tutorial. Can you hear me?
Yes.
Just want to emphasize that even though these are a little bit of context for the tutorials that come later, still should be pretty informative for all of our attendees today. So Stephanie?
Yes, so now we'll be hearing from our first tutorial speaker, Michael Thornton, and his talk will be on deep neural networks for assessing cortical responses to speech. So just a little bit about Mike. He's a PhD student at Imperial College under the supervision of Tobias Reichenbach and Danilo Mandik. Prior to this, he received his master's in physics at the University of Oxford. Mike is funded by the UKRI Center for Doctoral Training in Artificial Intelligence for Healthcare. And at Imperial College, he currently studies how neural processing of continuous speech is reflected in a listener's EEG. And in today's talk and tutorial, he will be covering how he uses neural networks to decode the speech envelope from EEG recordings. And he'll also look at the associated statistical analysis you can perform on your results. So Mike, do you want to take it away?
Yeah, sure, thanks a lot. Yeah, can you hear me?
Yep, and we can
Good.
see you and your slides.
Good stuff. Okay, yeah, so thanks a lot for coming to deep neural networks for assessing cortical responses. So I'm supervised by Danilo at Imperial College London and also Tobias at Erlangen. And again, many thanks to UKRI for supporting this work. And yeah, the CNSB organizers for giving us the opportunity and the initiative, which is going to be really helpful for data-hungry things such as these DNNs. So yeah, we've seen a lot of encoding and decoding models so far, so I won't do too much intro on those. Just a reminder, we have these these typical flavors. So the encoding models go from the stimulus to whatever modality you're looking at. So for example, the speech envelope to the EEG recordings. And we end up with these forward models. You get these temporal response function weights, which hopefully tell you something physiological. And we also have backward models where we essentially do the same thing in reverse. And we can try to reconstruct stimulus features, such as the speech envelope. And yeah, you'll see we get less exciting reconstruction scores with less invasive modalities typically, but we can also see that we can still get significant results if we try to ascertain the noise level of these reconstructions. So again, these are correlation coefficients. We've got this null reconstruction distribution, which tells you the noise level. We can see the actual reconstruction distribution seems quite different to it. And yeah, I'll cover a bit more of that in the tutorial if you're interested. So a bit of a precursor to why we looked at the DNNs. What's the assumptions that kind of go into the TRF model is firstly linearity. So we assume in the example I gave just then, the EEG response is assumed to be a linear function of some preceding values of the speech envelope. Then we also assume time invariance. So these coefficients are typically fixed. And in this kind of LTI system formulation, you get this nice property that the TRF coefficients represent the impulse response of the system. So this is why we can make, we can attempt to make a physiological insight because they effectively show how the brain or whatever modality we're looking at responds to this stimulus. So yeah, they're really useful. It might be interesting to start to play with some of these assumptions. So for example, you could try to break linearity. So you could use, well, in this talk, it'll be using nonlinear models, nonlinear backward models. There are other things you can do. So the speech envelope itself is a nonlinear transformation of the speech waveform. You can choose other nonlinear transformations and then do, for example, a linear modeling approach. And you can also attempt to break the time invariance assumption. So there have been some attempts in the literature to look at adaptive decoders or encoders, and dynamic TRFs, where you look at the temporal response function, not across the whole trial, but across much smaller temporal segments. And you can try and see if there are any dynamics there. But yeah, so here we're just going to look at nonlinear backward models. Before we dive into that, just a slide on where nonlinear decoding has been used in some related spaces. So if you type in nonlinear decoding of motor imagery, you get all kinds of stuff. So various modalities, people are interested in lots of stuff, such as prosthetic control, that kind of thing. There's a really nice example very recently about brain-to-text decoding from intercortical responses, and also an attempt at phenome decoding, which is pretty cool. But we're going to, this talk is kind of framed in the setting of auditory attention decoding from EEG recordings. So to explain what that is briefly, the left picture is this classic cocktail party scenario. The idea is that we all probably have this pretty strong ability to tune in to the speaker we're trying to listen to, and suppress or ignore other speakers. And it's interesting to look at this using forward models and see whether there's some modulation in the TRFs, for example. So that's in the middle panel. And you can also do backward models, which may be useful for actual applications. So if you can decode the focus of attention using, I don't know, some measurement such as EEG, then you might be able to enhance a hearing device. So on the right, I show one of the glued together EEG devices we use inside the ear canal. And indeed, we can actually decode attention using this wearable device, which is pretty cool. So yeah, to expand on that a little bit more. So what we typically do is we train a backward model to reconstruct the speech envelope of an attended speaker. So you can see in the top right figure, we've got this reconstruction of the speech envelope. And then we've got the attended envelope and the envelope of the unattended speech stream. So we can correlate the reconstruction against the actual attended envelope and the unattended envelope. And what you find is shown in the bottom left. So you find the reconstruction of the attended envelope is, in this case, always higher than the unattended envelope. So this is the trial-wise reconstruction score, correlation coefficient, and you find this is a marker of attention. So to move a little bit more real time, you could, instead of doing this across the whole trial, you could chop your data up into windows. So I've shown in the top right, I've chopped this data up into 2-second windows. And then I can compute a proportion of the time this attention marker says that the participant was listening to the speaker that we asked them to listen to. And we take this to be the attention decoding accuracy. So in the bottom right, you can see 2 examples for a 2-second window and for a 10-second window. Of course, the decoding accuracy is better for the 10-second window. The correlation coefficient is less noisy when you use more data. Okay, so that was kind of the intro matter. The rest of the talk is going to be about what deep neural networks are, then how we use these to decode the envelope in various scenarios, and how this leads to attention decoding. And the premise is that you might be able to get a higher decoding accuracy or something with DNNs as opposed to using linear models implemented via ridge regression. And then at the end, I'll just go through what's actually going to be in the tutorial to see if it interests you. So yeah, first of all, what are deep neural networks? I won't be giving a very comprehensive account of these, just because there are many resources online and I don't want to take up the whole talk, just talking about one algorithm. But the take-home message is are going to be they're heavily parameterized models. So they rely on a lot of parameters, a lot more than the linear models. And they are nonlinear. In fact, they can represent a lot of different functions. So you can call them universal function approximators. And that gives you some kind of guarantee that it's usually possible to find some function that does what you want it to do using a neural network. The question is then, how do you actually find the correct parameters? Once again, many resources documenting this. I want the take-home message to be that effectively you use calculus to maximize the performance of your model. So for example, you could calculate the mean squared error between your reconstruction of the attended speech envelope and the actual attended speech envelope. So yeah, I've shown this quadratic equation, which you could probably minimize fairly easily using calculus or whatever you want. For a DNN, it's usually not possible to do this analytically. So normally people use some flavor of these gradient descent algorithms. So effectively it's like a numerical algorithm for, or not necessarily numerical, but kind of an iterative algorithm for finding the minimum of these curves. Yeah. So what are deep neural networks? You'll normally see the FCNN in this figure, some variant of this diagram showing inputs being passed through layers of nodes. And once again, I'm not going to go too deep on that. It seems like quite a artificial thing, like kind of strange, but basically it's been shown that this is one way of producing a universal function approximator, which is something that we want. A note, just because I realize I picked bad acronyms for these two things. If you're familiar, the FCNN is a fully connected neural network. So the CNN in FCNN does not mean convolutional neural network, but the CNN for the second neural network does mean convolutional neural network. So sorry, but yeah, I hope that clarifies that. Yeah, if this was kind of mysterious, please just stick to the fact that they're nonlinear, they have many parameters, and we try to find their parameters using this iterative technique. So I'm gonna quickly go over some of the results that we found when we use these DNNs to decode the envelopes of speech. So we have two data sets. Our first data set has 13 participants. They listened to quite a lot of clean speech in their native language, 40 minutes. And in this plot, we had divided the speech, we had divided the data into 2-second windows, which is how we built up these distributions in these box plots. And yeah, we found the DNNs outperformed the linear method for most participants on the subject level and quite significantly on the population level. If you're interested in a different window size, the results are here. So yep, I've shown the mean of these box plots, where you can see this effect more clearly, the DNNs outperforming the linear method. And I've shown the variability or the standard deviation. And you can see the reconstruction score becomes considerably noisier for smaller window sizes. So this slide is about subject independent models. So if you're really interested in building some kind of smart hearing aid using ear EEG or something like that, then it's not really practical to sit your hearing impaired subject down for 40 minutes to record their EEG. At least our clinicians really just don't have time for that. So you might be interested in seeing whether it's possible to use a subject independent decoder, and the reconstruction scores, the correlation coefficients for those are shown in the middle group of box plots. You can see there's a big performance cut compared to the subject specific models. The DNNs do continue to outperform the linear model. You may need to consider whether subject specific information may actually be necessary, considering the difference in performance. our conclusions that the DNNs do still offer a considerable advantage. You may need to consider using subject-specific information. I've also used population models where a small amount of subject-specific information is included. So this lies in between the two extremes of all subject-specific information and no subject-specific information. So that was a good sanity check. Okay, so the last envelope decoding results slides, we used our second data set. This had more participants and they listened to some more interesting stimuli. So they listened to, again, English, which was their native language. They listened to Dutch, which was a language they didn't understand. They listened to English in speech-shaped noise, and they listened to competing speakers. So in this last case, there was a male speaker and a female speaker, and we directed the subjects to listen to one whilst ignoring the other, or vice versa. So you can see the envelope decoding results, and we found a considerable benefit of using the CNN, well, of using both of the DNNs compared to the linear model. For the foreign language, the results were all fairly similar. When we tried to reconstruct the envelope of the unattended speaker, we also found all of the methods performed quite similarly. But from these results, it looks like we can still do attention decoding just by comparing the attended reconstruction scores and the unattended reconstruction scores. So that's what we looked at next. So just a reminder, we are dividing the test data up into windows. We look at the reconstructions in each window. We try to see whether the attended envelope had a higher reconstruction score than the unattended envelope. And from these figures, we come up with a decoding accuracy. So yeah, I show the decoding accuracies for 3 windows here. So we've got a 2-second window, 5-second window, and a 10-second window. Once again, we see this relationship where the decoding accuracy increases with the window size, which again makes sense. We have more data, we have a more robust correlation coefficient. But also it looks like the DNNs are again outperforming the linear model quite considerably. And that should make sense based on the envelope decoding scores we saw earlier. So yeah, a summary of those results. We've mixed the female attended and male attended conditions together for this plot. And we also represent those results in a slightly different way. So we use this information transfer rate metric, which tries to take into account the window size. So we see basically, when you look at any one of these plots, you have to consider how long the window is and also how good the decoding accuracy is. And when you consider those things together for the using this ITR metric, you find that the 2-second window size seems to be the best. So that is consistent with the literature. There have been some attempts to do this. We've provided a more detailed, I guess, analysis of how well the actual envelope reconstruction aspect of this algorithm works. But there are a couple of other papers you can read, essentially using the same approach for attention decoding.
Hi, Mike. Yeah, I just want to give you a 5-minute warning.
Okay. Yeah, this is the last slide, so thank you. Yeah, so the structure of the tutorial is, yeah, in part 1, we'll go over fitting linear backward models briefly. And that's basically going to be important for part 2. So we want to really understand training, validation, and testing, and hyperparameters. We're also going to begin to look at testing the reconstruction scores for significance. In part 2, we're going to fit some DNNs. We're going to look at how do you stop this iterative gradient descent algorithm using pruning. We're going to look at how you use Optuna to tune the hyperparameters. and how do you actually evaluate the DNNs? And then hopefully part 3 will be more interactive. I'm hoping we'll be able to analyze your results and actually compare the DNNs against the linear models and see which are better. So I'm happy to share the slides. So if you're interested in the references or anything like that, that's okay. And that is the end. Thanks.
Great. Thank you for your talk. Mike, would you like to answer some of the Q&As? The questions in the Q&A window?
Okay, found it. Is this a smearing effect? Yes. I think it's also to do with the regularization. Sorry, Vishal's question was that he sees the bump in the TRFs arising before 0 seconds. He wants to know if this is to do with my filters. I would say yes. I'd say I've low pass filtered these data below 8 Hertz, I think. I think I low pass filtered the EEG, maybe the stimulus. But also I've used ridge regression, which smooths out these TRFs. So what may have been a sharper peak will actually be more spread out as well. Are they CNN or FCNN trained from scratch, or are they transfer learning? Yes, they are trained from scratch. Yeah, so we had some difficulties with the second data set because there are only 10 minutes of data per condition. And this is why I said down here, we had to mix several conditions to get enough data. And we found that we needed about 20 minutes of data to actually make any progress here. So yeah, that's probably related to the quality of the EEG data set, basically. Yeah, that's why we needed such big data sets. Was the data set using scalp EEG or in-ear EEG? Yeah, we used scalp EEG. You can see some studies use, at least one study using ear EEG. We're collecting an ear EEG data set ourselves. But yeah, the results were using scalp EEG. Things look a lot worse, as you'd expect, with ear EEG.
Yeah, I have a question if I can.
Okay.
Yep.
I mean, it's more of a, I don't know if it is a criticism or more of a question in this case. Ridge regression, I love it for forward models, but it's certainly not the state of the art in terms of decoding of attention, for example. So what I use in the past and what Daniel Wong and Anand Shmini use is CCA. So I would find a comparison, a more appropriate comparison with CCA rather than ridge regression. Do you have that? Do you plan to do that?
I plan to do that because the same criticism arose the last time I gave a similar talk. I think I've got the reference somewhere. There was this review by Seaman where you can see some curves comparing window size and decoding accuracy. And actually, the CCA was the best, but it didn't, at least for the 2-second window, it didn't come out as the performance boost over ridge regression wasn't as high as I expected. When they used larger windows, sure, it was a lot better. But yeah, I agree that would be a valuable comparison, especially on the same data set, because comparing between different data sets, as you're aware, that's not ideal.
Mick, for the sake of time, do you mind saving your question for 5:30 once we have our break between the tutorials?
Yeah, sure.
Great. Thank you again, Mike. So now we're going to have Joshua. Joshua, do you mind sharing your screen?
Sure. Yep. Is
Great.
everything okay?
Yep, we can hear and see. I'll just introduce you now. Okay, so now we'll be hearing from our second tutorial speaker, Joshua Kulasingham. He'll be giving us a talk on algorithms for estimating TRF components from MEEG responses to continuous speech. So Joshua is a postdoc in the Department of Electrical Engineering at Linköping University, Sweden. Prior to this, he performed his PhD at the University of Maryland College Park under the supervision of Jonathan Simon. His thesis was entitled Time-Locked Cortical Processing of Speech in Complex Environments. Now he uses EEG methods to study neural processing of low-level and high-level features of continuous speech. In today's tutorial, he'll cover algorithms for estimating TRF components using EelBrain, Python toolbox for MEEG encoding/decoding analysis. So Josh, take it away.
Okay, thank you for that introduction, and yeah, thanks for giving me this opportunity to talk about this topic. Yeah, so this is going to be more of a methods-heavy talk. It's going to be about the different algorithms that people can use to estimate TRF components. And by TRF components, I mean these peaks that we normally see in typical TRFs or encoding models. So I'm currently with Linköping University, but this work was done during my PhD at the University of Maryland with Jonathan Simon. Okay, so to motivate this, let's first consider the evoked response. And a TRF can be thought of as an extension to an evoked response. So here you have your traditional evoked response model where you have a stimulus presented and you have some response. Usually it's much more noisier, and so then you average and you get a response, and that response has some components. I think many of us are familiar with these, you know, the P1, N1, and so on. So one way to think of the TRF is as an extension to this. So if you have impulses that are scaled, you get evoked responses that are scaled. And if you have impulses that are close together, then you get evoked response that are overlapping. And so if you have a continuous predictor like the speech envelope, you can think of an evoked response to every point of this speech envelope that is scaled and overlapped together to give you your response. So this actual evoked response would be the TRF that we calculate. Now, this is not the only way to think of the TRF. I think there have been other interpretations of the TRF in previous talks. But if we consider it in this way, then it's interesting to think about these components. And is there any sort of match between the TRF components and the evoked response components? And so that's usually what we see in TRFs to the speech envelope. We typically see a sort of a peak like a P1, N1, and a P2. And in my work, I'll be using MEG. So I'm going to use the MEG terms, which is the M50, M100, and M200. So like the 50 millisecond, 100, and 200 millisecond components. Now, the interesting thing about these components is that they change based on the task. So for example, when you look at people listening to speech and when they attend to the speech, you see a difference in this component here compared to when they don't attend to the speech. So you can interpret this as perhaps the earlier processing stages don't really have attentional modulation, and then later processing stages have some attentional modulation. And so usually when we do this sort of analysis, we would look at component amplitudes and component latencies. Another example is when you have older and younger subjects. Older subject TRF components are much higher amplitude compared to younger subjects. So both of these types of effects have been seen in many studies, and there are several other types of effects that you could see using these components. Now, all of this is to group average data, but when you look at individual TRFs, they're often quite noisy, right? And for example, in this particular individual TRF, you don't really see very clean components. Sometimes when you have sensor space or source localized TRFs, where every line here is a TRF at a source or a sensor, it's even worse. You can't really detect these components very well. So our question was, is there a way to estimate more robust TRF components? And which algorithms are the most suitable for this particular task? So in prior work, there have been some studies that looked at comparing forward models and sort of looking at how they perform in terms of model fits, reconstruction accuracy, correlation, and also these studies looked at things, regularized models and some nonlinear methods like DNNs. So in this work, we want to compare algorithms based on TRF component estimation, not just on the correlation coefficient. And we also want to compare two widely used algorithms, the ridge regression that I think everyone here has heard about, and also the boosting algorithm. So the difference between these two is that ridge is smooth and boosting is sparse. And I'll talk a little bit more about that later on. We also want to see if we can find some sort of new method that would utilize the prior knowledge that we have about these TRF components, specifically to the speech envelope. So by this prior knowledge, what I mean is that we would expect a component in this window, another component in this window, and another component here, let's say. So can we use this sort of latency windows and try to find these components by directly estimating their amplitudes and their latencies? So to go into these algorithms that we looked at, we have ridge regression and boosting, and then we also have two other algorithms, subspace pursuit and expectation maximization. And these two algorithms are going to be using this prior knowledge to directly estimate the component amplitudes and waveforms. So we tested these algorithms using simulations and real MEG data on single channel sensor space and source space TRFs. And we also looked at their TRF component estimation accuracy. So now I'll just go briefly over each of these algorithms and just give you an idea of what they do. Oh, and by the way, this work is now published, so please go ahead and look at the paper if you want more details. So ridge regression, I think we are quite familiar with this since I think many people here have been using ridge regression. So this is the ridge regression TRF model where you have your EEG or MEG response, and then you have a shifted predictor matrix. So this would be the speech envelope that's been shifted by the time lags of your TRF, and this is your TRF beta, and this is the noise in the system. And we have the ridge solution with this regularization parameter. Boosting takes a slightly different approach. So here boosting is actually an incremental method, and it starts with an all zero TRF. And we want to build up our TRF by adding small values to the TRF, right? And these small values are not actually impulses, they are some smooth shapes. And when we construct these TRFs by adding these values, we have a predicted signal, and we compare that predicted signal with our training data. And then we choose the best change and keep that in our TRF. And then again, we predict the data and then compare it with a validation data set that's separate from the training data and see whether the error is improving. And if it does improve, we keep continuing this loop and build up our TRF until the error stops improving. So it's an iterative approach, right? So since this stops as soon as the error doesn't reduce anymore, boosting leads to more sparse TRFs. So this is a ridge regression TRF at the top and a boosting TRF here. And this was actually estimated on the same data. And you can see that although the peaks are in the same, like the similar latencies, they are very different in terms of the shapes of the peaks, right? Some characteristics of boosting is that you don't need to tune the regularization parameter because there is no regularization parameter. So we heard about things like banded ridge, where you may have to have different regularization for different predictors. But in boosting, you can use impulse predictors and continuous predictors together without worrying about that regularization. It also has less spurious activity and clearer peaks. So typically you are able to see some nice peaks in boosting TRFs. But at the same time, boosting may also miss some smaller components. And it may not show you all the subtle variations in the TRF if you are interested in that, if you're interested in some peaks that are very small. So in the tutorial later on today, I'm going to go into the EelBrain toolbox, the Python implementation of boosting. And I'll be talking about how you can use this EELbrain toolbox to look at MEG or EEG data and how you can fit this boosting TRF. So if you're interested in that, please, you can come to the tutorial. And then we also have a proposed algorithm. So these algorithms are the ones that are going to use the prior knowledge of the TRF components. So the TRF model for these algorithms is slightly different. Instead of a single TRF, we have different components. And these components are shown here. So there are J components. In our case, maybe there are 3 components for the 50, 100, and 200 millisecond peaks. And these components are shown here in CJ, and they each have an amplitude here. And we want to directly estimate these two. And the way we do that is using this subspace pursuit algorithm. So first we define a smooth component shape or a basis. And then we find the best basis within a particular latency ranges for each of the component. So we want to find the best basis here, here, and here. And we also estimate the amplitudes of those components using least squares. And then after subtracting out the fitted components, we iterate this procedure until we converge and the error doesn't improve anymore. So this will give us a TRF that is comprised of only these 3 components within these 3 latency ranges. So this is for a single channel TRF. Now we can extend this for a multi-channel TRF model. Typically, multi-channel TRFs with ridge or boosting, they would use an independent TRF at each channel. But here we're going to use, we're going to fit the multi-channel TRF jointly. And the way we do that is that we have a spatial topography instead of an amplitude for each component. So each component has its own spatial topography, and that's going to give rise to our multi-channel response. And for this, we use this expectation maximization algorithm. So the expectation maximization has 2 steps. We have an expectation step where we estimate these spatial topographies. And then we have the maximization step where we estimate the latencies. And this step is actually very similar to the previous algorithm where we incorporate our prior knowledge of what these latency ranges are for each component. So using these algorithms, oh yes, and we repeat this as well until we converge. So using these algorithms, we looked at 2 different data sets. So we looked at a simulated data set with 30 simulated subjects, and they had ground truth TRF. So since it's simulated, we know exactly what the TRF components are. And we looked at single channels and sensor and source space TRFs, and we added some realistic MEG noise at several SNRs. And then we also looked at their performance on real data. So this was on a previously published data set, MEG data with 40 subjects, and they were listening to cocktail party stimuli. So a foreground and a background speech. So how do the results look like? So this is the ground truth TRF for a sensor space simulation. So this is the TRF for one subject. And since it's a simulation, we had this nice clear TRF with 3 components. And this topography that you see here is for the middle component, the 100 millisecond component. This is a typical MEG topography for the 100 millisecond component. And when we fit our algorithms on these simulations, we found that these were the results. You see that all of them are able to sort of get the same type of components, but there are some differences. For example, if you look at the subspace pursuit, it's much noisier. So subspace pursuit, boosting, and ridge fit TRFs independently at each channel or sensor, whereas EMSP is fitting the TRF jointly at each for the entire sensor space. And that's why you get this nice clean components for the EMSP. So yeah, so in summary, it seems that boosting, ridge, and EMSP are giving comparable results. Boosting and EMSP are sparse. EMSP is sparse by design because it has to fit only 3 components. And ridge and EMSP have better topographies. So to further quantify this, we looked at some performance metrics. So the first type were the model fits. So here we have our response correlation. This is what we have been talking about for many of the studies that we've heard so far. This is the correlation between the actual and the predicted response. And then we also have our TRF correlation. Since we have the ground truth TRF, this is the correlation between the ground truth and the estimated TRF. And in both these cases, it seems that EMSP is performing better over the different SNR levels. When we looked at the other metrics, we have a different story. So here you have component estimation accuracy in terms of latency error, amplitude error, and topography errors. And here you see that there is a much larger spread, and it seems that many of the algorithms have comparable performance. Interestingly, Ridge and EMSP seem to do better in terms of amplitude and topography errors. But also we looked at another metric of spurious activity. So in the ground truth TRF, there was no activity after 300 milliseconds. But in our fitted TRFs, especially for Ridge and boosting, we have some activity there. So that we consider that as sort of spurious activity. And you can see that Ridge has more spurious activity than boosting, which isn't that surprising since boosting is supposed to be sparse. When we look at our source space simulations, we have similar results. So this is the ground truth for source space. We looked at this region of the temporal lobe. And you have the fitted TRFs for each algorithm. And they look very similar to what we saw in sensor space. And finally, let's look at real data. So this is the fitted TRFs for a real data subject. You see that firstly, that real data TRFs are much noisier. And you can see also that the boosting is sparse. Ridge is much smoother with a lot more activity. And EMSP and SP are able to detect components that are similar to what boosting and Ridge are also detecting. In terms of the topographies, it seems Ridge and EMSP are doing better, whereas boosting and SP are much sparser. So in terms of the metrics that we can use for real data, since we don't have a ground truth on which components have to be estimated, the only metric we have is this correlation or different measures of the error. So we use the correlation, the response correlation. And you can see that across subjects, there's a wide variation, but all the algorithms are comparable, except that boosting has slightly lower correlation values. So you can see that over here. And that may be because boosting also is sparser, and so you don't have a TRF for many of these sensors. But in terms of the variation, you can see that there is a large amount of individual variation. So in conclusion, EMSP seems to perform well in simulations. It has the best model fits and the best component estimation, but not as well on real data. So this may be that we don't have proper assumptions on the latency windows, or maybe there's just too much individual variability. Perhaps some subjects don't have those components because of anatomical reasons or because the noise was too much. So there has to be some more investigation as to how we can use these EMSP in real data and what type of assumptions we need to build into it. As for ridge regression and boosting, ridge regression had slightly higher model fit correlations, but it also has spurious activity, whereas boosting, you get these nice clear peaks, but it results in lower model fit correlation. So again, for more details on these algorithms and this comparison, go and take a look at the paper. And with that, I'd like to end by saying thank you for listening, and thank you to my advisor and the members, my lab members at the University of Maryland. And just very quickly about the tutorial. So the tutorial is going to go into the EELbrain toolbox, and it's going to be an introduction to how you can use the EELbrain toolbox to look at TRF. So it's going to talk about everything from loading the files right up to statistical tests across subjects. So if you're interested in that, see you later today. Yeah, so I think we can open it up for any questions.
Thank you. Do any of the participants have questions that they weren't able to type into the Q&A box? Aaron?
Hi, Joshua. That was a great talk. I'm particularly interested in your EMSP algorithm. Recently, not so recently now, Jack Gallant and Alex Huth put out a paper where they talked about ways of enforcing some prior knowledge, and they did it by changing, I think it's by changing the regularization function. So usually in ridge regression, we use like a diagonal, and then you scale the diagonal. And what they do is they use, instead of just a diagonal, they can enforce a smoothness prior where they've got, have you seen this paper?
I think I may have, yes, but
Okay.
I can't remember all the details. Yes.
It's actually where I got the banded ridge code or from. So I was curious if you've compared your solution to it, because it seems to be doing the same thing, except that you're sort of just fitting the TRF itself. They're sort of enforcing this on the regularization. And if you haven't, maybe could you like maybe speculate on like how they might be different or what they, you know?
Yeah, I'm not sure if they are exactly the same conceptually,
Okay.
because when you change the regularization, I mean, I'll have to look at their method in more detail, but what I can understand is when you change the regularization, maybe you have differences in the way that you are taking into account neighboring samples of your stimulus, right?
Yeah.
And the autocorrelations in between them. But you're still fitting a TRF without any restrictions on how many components the TRF can have or what particular latencies your TRF should have those components in. Whereas here, this is more sort of strict in that way that you are enforcing that it has to fit these components within these latency ranges. But you're right. I did not look into more complicated methods for estimating TRFs. I just focused on the basic ones like ridge and boosting. So maybe those would even do better than ridge in these comparisons. Yeah.
Cool. Thanks.
Nate, did you have your hand up earlier?
Yeah, I did. I can ask a question. So I was wondering, in the EMSP algorithm, I might have missed it, but I was wondering how you're making an assumption about the spatial distribution. Are you like assuming a certain spread of the spatial distribution? How does that work?
No direct assumptions. It's actually a maximum likelihood estimation of the distribution. So it's estimating the distribution.
So it does that for each channel independently?
No, it does it together.
I guess that is part of my question. I would have expected that there's some underlying distribution that that's part of the likelihood. Yeah.
Yeah. So it uses a Gaussian prior, but that's like a Gaussian for the prior distribution. But after that, it just estimates it from the data from the maximum likelihood. Yeah.
I mean, do you have any other thoughts on the type of spatial distribution that might help better fit the real data?
I'm not sure about that. So it was just an open prior distribution that is sort of flexible enough to fit to the real data. So a Gaussian would give you sort of a least square type of likelihood. So if you want something more like different, maybe you can use different distributions, but I haven't thought about that specifically. Yeah.
Cool, thanks.
I have a quick question. I'm wondering if you've been able to analyze Jonathan Simon's 2017 MEG dataset that had 3 talkers using this method. I
No,
just love that paper when I found it a couple of years ago.
No, I haven't had the chance to do that, but yeah, you're right. It would be interesting. Yeah.
We talk about that paper all the time in our lab.
If there aren't any other questions in the moment, I'd like to conclude this session. And anyone that has any unanswered questions for both Mike and Joshua, you feel free to stick around. I cut people off for the sake of staying on schedule. And anyone who signed up for the tutorials, please find those Zoom links in your inbox, and those tutorials will start at 6 p.m. Mick, would you like to ask your question? For Mike?
first of all, thanks Joshua and thanks Michael for the talks. They were great. To Michael, I had a quick question. I was just wondering if you saw any differences in the performance of the fully connected and convolutional neural networks, given that you seem to compare both of them to the linear model every time. Would you have expected to see better performance in the CNN, given the multivariate nature of the input to the models? And the other thing I was wondering was, for the fully connected models, did you have to extract temporal features from the data before you input them to your model?
Good questions. So the reason I liked the FCNN, which was proposed originally, is because it's kind of the simplest thing you can do. So I find with a lot of this stuff, like you can add on loads of bells and whistles and then you don't know what's important. So we wanted to just compare this to one other thing. And in fact, we found not only were the performances very similar, maybe, I think it makes sense. The predictions were also extremely correlated. Like we were seeing R in excess of 0.9. So yeah, we thought, we were fairly convinced that they were learning to do the same thing. So yeah, those are similar. The second question was about extracting features. No, we didn't extract any features. I mean, in principle, we didn't really need 64 channels, or I guess 63 channels referenced. So we could have reduced the dimensionality. But in fact, we found for both DNNs, we needed about 20 minutes of data, and just using the speech envelope and the raw EEG recordings or filtered and stuff, they worked. They worked when we had that amount of data.
So was the fully connected model able to relate EEG at certain time points to different time points in the stimulus, the way you had it set up, or was it a one-to-one mapping?
Okay. So are you asking about kind of single lag decoders, that kind of thing?
Yeah, essentially. Yeah.
I did actually have a go at that, because of course we wanted to have a go at like see if we could extract anything sciencey from these things. I need to have another go at it because the results, when I was doing before, the results were very different to the, for instance, the single lag decoders you get using linear methods. So I was seeing kind of a slow drop off rather than bumpy stuff. But like, it wasn't the most systematic analysis, so I don't want to make a claim that the linear model's predicting something different. The DNN's predicting something that's physiologically different to the linear model. But yeah,
okay, so the way you had it set up, you were able to relate information at different time lags to the stimulus, correct? It wasn't a single lag type mapping.
Yeah, so the results I showed were like 400 milliseconds of EEG after the stimulus. Yeah.
Cool. And you had to extract those features yourself, those temporal lags yourself, before inputting it to the model.
As in, we came up with this kind of matrix of shifted. Yeah.
Cool.
We had to,
cool. Yeah. Thanks.
Okay, thank you.
Do any of the participants have any other questions? If not, we can close this Zoom so the tutorial speakers have a break, and then you can ask those questions during the tutorial.
Suppose I have a very quick question if Joshua is still around, but I don't know. Oh, you're still there, Joshua. I thought you were having a break before the tutorial. Very quick. How did you select the optimal lambda in the TRF models?
Oh, using cross-validation. Yeah.
But did you just get the highest value?
Sorry?
Did you just get the best lambda, the highest value? The ones with
yeah,
highest
yeah, the
prediction?
best, yeah, the best, yeah, the lambda that they perform the best across the folds. Yeah.
Okay. Yeah, I mean, this is partly, you know, there was a question earlier on during Michael's presentation. It was from, I can't remember now, Bisham, I think. He was asking, you know, why does the TRF start rising before 0? And of course, like the thing is that when we look at the temporal response functions, generally we should be considering the peak of the component as, oh, that's the timing of the component, exactly because the reach TRF has so much smoothing potentially, you know, depending on the lambda. And just that I found also just in the last couple of years, I don't tune using the best, the lambda with the best prediction correlations anymore, just because the tuning curves often have this characteristic where they rise and then they kind of are almost flat, but they keep rising of nothing. And so you end up having lambdas that are 1,000 or 10,000 times bigger than this other point, this other lambda, but maybe the prediction correlation is 1% higher. And so it's not worth it. So I kind of use this thing where I set a limit. It's kind of like the AIC or BIC approach for computational complexity. It's not the same, but you know, I just set a limit of 1%, and when the lambda stop, the prediction correlation stops increasing, I stop there. So that helps a little bit. It's still not as good as boosting in terms of recovering the actual shape. I still find it funny how the prediction correlations are higher for ridge while the shape is not as well recovered for those particular cases. So it's just a funny one.
Yeah,
like your results are cool. I'm still, you know, the output sounds to me like, oh, I'll keep using ridge and you keep using boosting. You know, there is no best thing to do. It depends on what you want to do.
I think yeah, that's one of the problems with you know, trying to figure out these complex things. But yeah, like boosting, I think has its own biases. Like you said that boosting maybe is getting the best shape, but I mean, boosting is sparse and we give this sort of basis shapes, right? And so depending on that, the width of the component changes a lot. So it's not that boosting is, you know, I'm not sure that boosting is the, you know, the correct shape or what is the correct shape. But definitely it's easier to see the components with boosting, I think. So that's what I would think is good. But
so let's consider a case that is the opposite of sparsity. You know, let's say that you have the M400 that maybe has two components, just one after the other, which becomes a sort of long response. Would boosting tend to make it smaller than it actually is, because it's kind of forcing sparsity?
That's a bit hard to say. I mean, if there is enough, I mean, it just depends on if the error is you know, improving enough, right? Because boosting can easily continue to fit for a long time as long as the error improves. So if you give it like small enough basis, you know, basis dictionary, then you may get two components instead of just one smooth component. So yeah, I mean, yeah, like there are some parameters that have to be considered, I think, if you want to look at that. Yeah.